Hello friends, so in this video we are going to discuss about physiology of bacteria. First we will discuss bacterial growth and nutrition, then factors affecting growth. So starting with the first point, that is bacterial growth requirement. Okay, so water constitutes about 80% of bacterial cell. We have discussed previously also this point. Okay, 80% of bacterial cell consists of water. The minimum nutritional requirement that are essential for growth and multiplication of bacteria include sources of carbon, nitrogen, hydrogen, oxygen, and some inorganic salts. So, those are very in small amounts, such as sulfur salts, phosphorus salts, and other elements like sodium, potassium, magnesium. Okay, so these are the basic requirements of the bacteria. Now coming to the bacterial vitamin. Some fastidious bacteria do not grow in the routine culture medium unless certain organic compounds that are essential to those bacteria are added to the medium. These are known as growth factors or bacterial vitamin. So we have discussed bacterial vitamin or bacterial vitamin definition. So bacterial vitamin, again I am repeating, some fastidious bacteria which do not grow in routine culture. Those bacteria do not grow in routine culture unless and until they are given certain organic compounds. Clear? And those organic compounds are known as bacterial vitamins. In most cases, bacterial vitamins are same as the vitamins necessary for mammalian nutrition, particularly those belonging to vitamin B group. Okay, such as your thiamine, nicotinic acid, riboflavin, okay, pyridoxine, folic acid, and vitamin B12. So this is bacterial vitamin. Now the next point is bacterial cell division. So bacteria divide by a relatively simple form of cell division that is known as binary fission. We have discussed previously also about this. Okay, the cell division commences when a bacterial cell reaches a critical mass in its cellular constituent. Okay, so this is the basic principle behind the cell division. Now the nuclear division precedes cytoplasmic division. This is important. Nuclear division precedes cytoplasmic division. Clear? So first we will discuss two point about nuclear division. So the two strands of bacterial DNA are separated and they are replicated to form new complementary strands. Thus two identical molecules of double stranded DNA are formed. So this is about nuclear division. Cytoplasmic division, a transverse septum is formed. Okay, a transverse septum grows across the cell wall from the cell membrane, following which the cell wall materials are deposited and then two daughter cells are separated. Okay, so this is basic about division. We have discussed division in a separate video. Okay, so this is basic, most basic. Now we will discuss one important point about bacterial cell division, that in few bacteria the daughter cells may remain partially attached even after the cell division, so that bacterial cells are arranged in a pair or in chain. Example is streptococci. Okay, in a streptococci the daughter cells may remain partially attached even after the cell division, so the bacterial cells are arranged in a pair or in chain in streptococci, or streptococci arrangement is in chain or pair. If arrangement is in clusters, if arrangement seems to be in clusters, then that will be staphylococci. So in staphylococci the arrangement is in cluster form, and in streptococcus the arrangement in chain or pair form. Clear? So this is basic about bacterial cell division. Now moving to the next part, that is rate of multiplication. Clear? So first, in the rate of multiplication we will discuss one definition, that is generation time. So generation time is the time for a bacteria to give rise to two daughter cells. So here parent cell, from parent cells two daughter cells arises, and the time taking between arise of the two daughter cells from a parent cell is your generation time, and this time is measured under optimum conditions. Okay, now the generation time for different bacteria such E. coli generation time is 20 minutes. Okay, if you are looking for Mycobacterium tuberculosis, so the time will be 10 to 15 hours, and Mycobacterium leprae, the time will be 12 to 13 days here hours. Clear? No problem here. Now we will look for some types of bacterial count. So the first one is total count. It indicates the total number of bacteria, whether live or dead. Important, live plus dead. Count of bacteria is under total count. Okay, and this is done by counting the bacteria under microscope using counting chamber. The next one is viable count. It measures the number of only living bacteria in a specimen. It measures about viable count, measures about only living bacteria. Clear? And viable count may be obtained by many methods, which we will discuss later on. The power plate method, surface viable count by spreading method, or surface viable count by Miles and Mishra method, which we will discuss, okay, in next videos. Now we will discuss one important curve that is known as bacterial growth curve. Okay, so bacterial growth curve. So first I will make a diagram for the growth curve. Here number of cells, and on x-axis there will be time. Clear? Graph will be like this here. Okay, graph it divided into 4 parts. So the first part of the graph is lag phase. This is your lag phase. Okay, and the second part is log phase, and the third part is your stationary phase. Okay, and after stationary phase, suppose I am drawing the diagram for okay, this diagram is for total count. So this is log phase, and this is probably a stationary phase, like this. Okay, the stationary phase, and then will be continued, and this is for total count. If we are going for total count, then this graph will be there. If we are going for viable count, then after this there will be drop, because viable count only count viable means living organism, living microbes. Clear? So for viable count the graph will be going down, and for total count, if you are drawing graph for total count, then it will be like this. Clear? This is lag phase, log phase, stationary phase. Clear? Now we will discuss individually different phases. So first, coming to the lag phase. Okay, so lag phase is the period between inoculation and beginning of the multiplication of the bacteria. After inoculating into culture medium, bacteria do not start multiplying immediately, but take some time to build up enzymes and metabolite. Clear? And that is known as lag phase. Lagging, it is lagging. Then comes log phase. In this phase, bacteria, then comes your log phase. In this phase bacteria will divide exponentially, so that growth curve takes a shape of a straight line. Okay, takes a shape of probably a straight line. Clear? And in this stage, in this log phase, the bacterium is in characteristic of log phase. Bacteria is in a smaller in size. One more point, they are biochemically active, and it is the best stage to perform the biochemical reaction. Okay, log phase is the best stage to perform biochemical reaction. And the third characteristic is uniformly stained. Bacterium are uniformly stained. It is the best time to perform the gram stain, because they are uniformly stained. So we usually perform staining during this log phase. So log phase has 3 characteristics. Bacterium will be smaller in size, biochemically active, and uniformly stained. And characteristic of lag phase, where the bacteria increase in size due to accumulation of enzymes and metabolites. Bacteria reach their maximum size at the end of lag phase. So lag phase has 2 characteristics, and log phase has generally 3 characteristics. Then coming to the next, that is your stationary phase. Okay, so after the log phase, the bacterial growth ceases almost completely due to exhaustion of nutrients, accumulation of toxic products, and autolytic enzymes. Now, characteristic of the stationary phase are the number of progeny cells formed is just enough to replace the number of cells that die. Okay, this is the first characteristic. Okay, now in this phase, bacterium becomes gram variable also. In this phase, bacteria becomes gram variable. More storage granules are formed in this stage. Storage granules are formed because there is deprivation of nutrients, exhaustion of nutrients. Sporulation starts in this phase. In this phase, bacteria starts to produce exotoxins, antibiotics, like this. Okay, so these are the characteristics of stationary phase. Clear? Now we will discuss one more important point, that is one more phase is there, decline phase. If we are going for looking for viable count, then it is the decline phase. Clear? Now one more point I want to mention here, that is about culture. We will discuss in several videos also, but also here, that is continuous culture. So it is a special type of culture which supports the bacterial growth in a particular phase, that is log phase. Okay, for longer period, continuously, without depletion of nutrients or without accumulation of toxic product. So we use continuous culture to maintain growth in log phase. Okay, so this is the specialty of the continuous culture. Special devices are used which supply the nutrients and remove dying bacteria cells and their toxic product. Continuous culture can be maintained mainly by two methods. And the first one is your turbidostat culture, okay, and another one is your chemostat culture. So these two types of. Now we start with factors affecting growth of the bacteria. We have discussed about general physiology of bacteria. Now we are focusing on the factors. So there are several environmental factors that affect the growth of the bacteria. So starting with the first, that is oxygen. Okay, so on the basis of their oxygen requirements, bacteria is classified into a number of groups. So we'll discuss each of the group. So the first one is obligate aerobe. Okay, they can grow only and only in the presence of oxygen. Examples are Mycobacterium tuberculosis, very important example, Pseudomonas, Bacillus, okay, Brucella. So these are the four important examples in which Mycobacterium tuberculosis is the most important. So Mycobacterium tuberculosis, Pseudomonas, Bacillus, Brucella, they will survive only in the presence of oxygen. Now the next one is facultative anaerobes. So they are aerobes, but they can also grow in anaerobic conditions. Okay, and in this category most of the pathogenic bacteria comes, such as E. coli, Staphylococcus aureus. Okay, most of the pathogenic bacteria comes under facultative anaerobic groups. The third one is your facultative aerobes. Facultative aerobes means they are actually anaerobes but also grow in aerobic condition. Example is Lactobacillus. The next one is microaerophilic bacteria. Okay, they can grow in the presence of low oxygen tension, that is about 5 to 10% of oxygen. Name itself indicating micro means low. Okay, so they can grow in low oxygen amount, that is 5 to 10% of oxygen. Examples are Campylobacter and Helicobacter. Campylobacter and Helicobacter. Campylobacter and Helicobacter. The next one is obligate anaerobes. So they can grow only in absence of oxygen, as oxygen is lethal to them. The organisms which are coming under obligate anaerobes, it means oxygen is lethal to obligate anaerobic organism. Example is Clostridium. For Clostridium, oxygen is lethal. It can cause death of the Clostridium species. Clear? So once again I am revising: obligate aerobes, Mycobacterium tuberculosis, Pseudomonas, Bacillus and Brucella. Facultative anaerobe, most of the pathogenic bacteria, E. coli and Staphylococcus aureus. Then facultative aerobes, Lactobacillus. Then microaerophilic, contains 5 to 10% of oxygen. Example is Clostridium botulinum. Campylobacter and Helicobacter. Campylobacter and Helicobacter are coming under the microaerophilic, and obligate anaerobic consists of Clostridium. Clear? One more, that is aerotolerant anaerobes. They can tolerate oxygen for some time but do not use it. Example is Clostridium histolyticum. Okay, aerotolerant. Clear? Microaerophilic. So these two are for oxygen. Clear? Now the second requirement, that is carbon dioxide. So organisms that require higher amounts of carbon dioxide for growth are known as capnophilic bacteria. Okay, examples include Brucella abortus, Streptococcus pneumoniae. Two examples you have to remember in this: Brucella abortus and Streptococcus pneumoniae. Now the third is your temperature. So most of the pathogenic bacteria grow optimally at 37 degrees centigrade, that is human body temperature. That is, however, the optimal temperature range varies with different bacterial species. Okay, and accordingly bacteria can be grouped into 2 or 3 groups. The first one is your psychrophiles. Okay, these grow best at temperature below 20 degrees centigrade. Examples are most of the saprophytes. One example I will mention here, that is Pseudomonas. Clear? Now the next group is psychrophiles. The next one is your mesophiles. So these grow within a temperature range of 25 degrees centigrade to 40 degrees centigrade. Clear? And examples are most of the pathogenic bacteria. So most of the pathogenic bacteria belongs to mesophiles. Meso means medium range. The next one is thermophiles. Okay, these bacteria grow at higher temperature in a range between 55 degrees centigrade to 80 degrees centigrade, and example of thermophiles is Geobacillus and Geobacillus stereothermophilus. Okay, this bacteria is also used in indicators for sterilization purpose. Okay, Geobacillus stereothermophilus in autoclaving. So psychorophiles, then mesophiles, then thermophiles. So this is the temperature based study. Now the next requirement is pH. Okay, so most pathogenic bacteria grow between pH 7.2 to 7.6. Very few bacteria such as lactobacilli, okay, can grow at acidic pH below 4. Lactobacilli can grow in pH below 4, while bacteria such as Vibrio cholerae are capable of growing at alkaline pH. So you remember lactobacilli in acidic pH and Vibrio cholerae in alkaline pH. So it is important. Vibrio cholerae can be grown in alkaline pH. Clear? So we have discussed oxygen requirement, CO2, temperature and pH. We will revise once again, okay, after doing all this. The next requirement is light. The next is your light. So bacteria except phototrophs grow well in dark. Okay, so light is not required so much to the bacteria. They are sensitive to UV rays and other radiations in light. Clear? Now one important point: photochromogenic mycobacteria produce pigments when they are given exposure of light. So light is not much required. Clear? Now the next one is osmotic effect. So bacteria are able to withstand a wide range of external osmotic variation because of the mechanical strength of cell wall. Bacteria can withstand variation in their osmotic gradient. Clear? Next important point: now sudden exposure of bacteria to hypertonic solution may cause plasmolysis. Okay, and this occurs more rapidly in gram-negative bacteria, because gram-negative bacteria has less amount of peptidoglycan in compared to gram-positive bacteria. The next one is that sudden transfer of bacteria from concentrated solution to distilled water may cause plasmoptysis, not plasmolysis. Here plasmoptysis means excessive osmotic inhibition of water leading to swelling and rupture of the cell. So there are two conditions: plasmolysis and plasmoptysis. Clear? Now we will move for the next, that is mechanical and sonic stress. Okay, though bacteria have tough cell wall, they may be ruptured and disintegrated by vigorous shaking with glass beads and exposure to ultrasonic vibrations. Okay, this is not so much important. Okay, now coming to the next one, that is moisture. So moisture is an essential requirement for the growth of bacteria, because 80% of bacteria cell consist of water. 80% of the bacterial cell consists of water, so moisture is an important factor. Okay, and drying has varying effects on different organisms. I will give one or two examples here. Some organisms like Treponema pallidum, Neisseria, gonorrhea, okay, die quickly after drying, while these two die quickly after drying. Okay, while Mycobacterium tuberculosis and Staphylococcus, these two, Staphylococcus aureus, may survive drying. Okay, it will survive drying for several weeks. Clear? So Treponema pallidum and Neisseria gonorrhea die quickly after drying, and Mycobacterium tuberculosis and Staphylococcus aureus withstand drying for several weeks. Clear? So this is about the factors affecting the growth of the bacteria. Clear? So thank you for watching. Best of luck.